1203. Let me just start by saying a couple things about the perennial sunflower program. So Kevin Betts and I had this discussion, it's probably 15, 20 years ago, about the idea of developing a perennial sunflower. And then we had a graduate student by the name of Brent Hulke that came in and played a key role in initiating this project, and really was part of the team that got the program off the ground. I don't see his name here on the list today, but just want to recognize that Brent Hulke was a key player in developing the perennial sunflower program, and he was working with me and a couple other folks here on campus on winter hardiness in perennial ryegrass, but he was using the perennial sunflower as a side project. And guess what happened? When he graduated, he ended up becoming the USDA sunflower breeder in Fargo, and he's still the sunflower breeder in Fargo today. So this gives you an idea of some of the experiences that the Forever Green Initiative provides, and I think Brent is a good example of some of the offshoots in terms of career development for a number of the students that come into the program. So it's a program that was basically kept together for a number of years by Kevin Betts, and he made a lot of progress and kept the program together until we developed a more advanced team, and it's more the advanced team that's going to be presenting today along with, I think, Kevin. So it's been a tough one. This has been a hard one to make a lot of fast progress on, but looking forward to the update from the team today. So what I'd like to do, I think you guys are organized. Jill, are you going to lead off?
Yeah, I'm going to start, and then Kevin's going to talk about the hybrids after me.
Okay, so I'll turn it over to you, Jill. Why don't you just give you a little background on your team and yourself, and we'll start from there.
Okay, so I'm Jill Ekar, and I'll be presenting for the first half, like I said, on the work developing perennial sunflowers. And so Kevin Betts and I are researchers who kind of have our hands on the projects. And then Bob Stupar and Yaniv Brandvain are the PIs. And our work continues to benefit from contributions from Mikey Cantar, who is now a professor at Hawaii Manoa, but he remains very involved in the work. So I'm going to be presenting kind of an overview of both of the projects that we've got going on, and then I'll give Kevin the floor to update you all on the hybrid sunflowers. And our team will be happy to field questions throughout, and I would just say go ahead and interrupt me, since I might be a little bit too hyper-focused on the presentation to notice any questions that pop up in the chat. And I don't see us probably taking up the full hour, so there should be plenty of time for Q&A at the end as well. Okay, so our group is working on 2 ways to achieve an agronomically viable perennial sunflower. And as with any perennial crop, the obvious benefit is that with a single crop, you can achieve year-round soil coverage without replanting. And then collect on all the ecosystem services that come with that. So to that end, we currently have 2 projects underway. We're 9+ generations into wide hybridization between the annual sunflower and tuberosus. And we're still in the very early stages of de novo domestication of divaricatus, but I'm excited to see where that project goes once we get the first couple of generations of crosses in the field and start selecting on their traits. So our goal in both of these projects is to select for idealized agronomic traits. So it's the ideotype or ideotype selection, however you say it. So in addition to perenniality, we like to have a plant that produces large seeds with harvestability traits like a large central head, and ultimately have a product that can compete with the annual sunflower in net economic gains. And the idea is ultimately for this to be a perennial oil seed, but there may be other markets for perennial sunflower, like bird seed. The birds definitely like my seeds. They're out in my field a lot right now. It was also recently suggested to us that perennial sunflowers could be value-added eco ingredients. So for instance, you could imagine these seeds in granola bars or products like that potentially. I'm going to start by talking about our efforts with our classic sunflower project, the one that Brent Hulke started with originally. So that's wide hybridization between annual sunflower and tuberosus, which is also known as Jerusalem artichoke. Our target plant looks like annuus above ground, much like the visual in the previous slide, but it will look more like tuberosus below ground. So we're selecting for perenniality, so tubers, big flowers, big seeds, fewer heads. In a lot of ways, these hybrids look really good already. So if you, when you see them in the field, you might even think they're annuus because you can see there are a lot of large singular heads here, which is great. And most of these plants also tend to overwinter. These hybrids have been intermediate in their traits, but with a lot of variability between them. And unfortunately, fertility has been a fairly long-standing issue with them. So we think that probably their low fertility results from uneven meiosis and fraught recombination between genomes of fairly distant relatives. So just to orient you with the subgenome notation, each letter here represents the subgenome of a different species. So tuberosus is on the left, and that's a hexaploid with 4 divaricatus genomes and 2 grossus serratus genomes or subgenomes. And those are both perennial sunflowers. And then the annual sunflower on the right is just a simple diploid. So F1s are auto-allotetraploids. And that means that during meiosis, when you have maternal and paternal chromosomes coming together to recombine, theoretically, the 2 A genomes should be pairing with each other and then the B and C genomes pair. But that second pairing may be occurring with varying levels of success due to the divergent ancestry of the perennial and annual sunflower genomes involved in that pairing. This here is a simplified pedigree of the various lines derived from our initial hybrid population. This is functionally a population since Kevin uses bulked pollen from favored plants, but you can see that there have been a lot of successful crosses in the RM4 background. And you'll notice that on some of our graphs, there are a lot of reddish orange dots, and that's because the dots on some of them are colored by maternal lines. So for instance, this plot in the upper left, so the x-axis on this plot is generations. So this is just showing that we have been successful in breeding for larger seeds over generations. So we are very happy about that. In 2018, our group found that perenniality in sunflowers is determined by only a few genes, which is good news for us because that means that selection for perenniality should be fairly simple. And this plot here shows perenniality over generations. And it looks kind of bad on the surface, like we're losing perenniality in later hybrids. But that's really just because individual plants lose perenniality or fail to overwinter. And we don't move forward any material that doesn't overwinter. So those plants are removed from our breeding population. But you can see in the other surviving individuals in like the 6th and 7th generation, that trait is actually, it seems to be getting stronger. So we don't see this as a real barrier with continued selection. We've also been targeting a plant with a larger central head rather than many small heads. In these scatter plots, again, they're both showing generational progress, even though both trend lines seem to be going in the right direction. So on the left, we're seeing slightly larger heads over generations, and on the right, we're seeing slightly fewer heads over generations. The trends are in the right direction, but that progress is not really significant. So we need to continue selection on those traits. And I should also point out that our most recent generation, or maybe two, they haven't been incorporated into this story because Kevin is still compiling those data for us. So in analyzing our data for a 2019 publication, we did discover that annuous traits tend to increase together. So head size, seed size, and apical dominance, they occupy the X and Y axes here to compare all three against one another. And you can see that they all show positive correlations. So plants that have larger floral discs also tend to have bigger seeds with fewer flowers, all of which are good. The bummer here is that when you take those same traits, those three traits, and compare them with perenniality, there is a negative correlation. So each of those three desirable above ground traits is compared to perenniality in this figure. I do want to point out that although we like to present a range for perenniality, this is based on Kevin's qualitative approximation. So any non-zero perennial index here implies that that plant had some overwintering success. So plants on the far left of each panel those are the ones that failed to overwinter, but all of the plants from the middle to the right had some overwintering success. So the picture is not as dismal as it may look on its face. And also notice that there are high levels of variation in those surviving plants too. So, and we select on that variation. So there are always plants that are high in annuous traits while also being good enough at overwintering. This is showing ploidy across generations, and we did find that ploidy was fairly stable over time, mostly hovering around the tetraploid condition. But unfortunately, a tetraploid genome was not a guarantee of functional fertility as we had hoped. So we're looking into what's going on with our pollen and our head filling to help us sort out the fertility issues. So these are stained pollen grains, and the cytoplasm with all the DNA in it will take in pink stain, and then the outer coating of each grain stains blue. So if there's no pink in the center of the grain, we assume that that's empty and inviable. So annuous pollen is on the left, and then there are two hybrid samples on the middle and right, and you can see that in the middle photo, the hybrid pollen grains are much more variable in size. And then on the right, there are many, the grains are really uniform in size, but a lot of them don't seem to contain any cytoplasm. And we did discover that hybrid pollen that looks like the right panel is more likely to correlate with higher female fertility. And by that, I mean head filling. So apparently filled grains in the right panel end up being better dads than the irregularly sized filled grains from the middle panel, if that makes sense. So cytoplasm presence is no guarantee of actual fertility. So we think that that's likely due to the genomic structure that's predicting whether meiosis will work. In particular, meiosis between those B and C subgenomes that I talked about earlier, where the annual and perennial genomes should be pairing and recombining, but there is significant evolutionary space between them, so their genomic structure could be different in ways that matters. if those chromosomes frequently have trouble pairing, you could end up with cytomixis, aneuploidy, unreduced gametes, or knots of chromosomes even, which may explain what's going on with the middle panel. We have ideas though, and we feel good about our new direction, and Kevin will talk more about this when I finish, but just briefly, because we do have many individuals with desirable traits, Kevin is going to be shifting to a strategy of creating unique self-compatible lines, hoping that isolating selfing lines will purge non-selfing variants from those lines. But I'm also hoping that this strategy could kill two birds with one stone, because I see fertility as our biggest hurdle, but selfing over generations could potentially also stabilize other genomic segments that may be causing trouble during meiosis, by also purging segments that are problematic in that specific line. So in essence, where crossing different lines could be introducing new meiosis hurdles every generation, with selfing, each line could potentially just overcome its own inherent genomic inconsistencies and potentially, or hopefully, overcome fertility issues faster. So yeah, Kevin will talk a little bit more about that later after I give a quick update on our other project. So that leads me to divaricatus, and divaricatus is commonly known as the woodland sunflower. So some benefits of this species is that it's genetically diverse, it's got a stable genome and consistent perenniality. So breeding and crossing should be fairly straightforward. And in crossing all the accessions reciprocally, so far I've only encountered 2 accessions that were not fully interfertile. And that was only in one direction of that reciprocal cross. And I'll likely be adding at least 1 additional accession to the field this next spring too, since I recently found more seeds in the lab. So this is just walking through the woodland sunflower field this summer. And it might be hard to gauge, but it's pretty non-uniform. It looks like there's a lot of diversity here to work with. So our idea is to cross the most promising plants with each other and then cross all the different accessions with each other to look for any signs of hybrid vigor. And I've also been working on doing all the crosses within a single accession so we can compare the phenotypic variance between SIBs and half SIBs, and that'll help us assess the diversity within an accession. I tried to zoom in on a couple of beetles here too, just to show that the pollinators also really like these sunflowers, as well as the birds, which come around a little bit later in the summer when the seeds are ready to eat.
Hey, Jill,
So,
would
yeah.
you tell them why we actually started to look at this species? What the original idea was in terms of using it to cross with annual sunflower, because it was a diploid, right? Could you just say, just so they understand,
Yeah.
we didn't go to domestication of this species. It was the decision that was made later.
Yeah, we had initially wanted to cross this with Annuus, and I spent a summer about crossing, pollinating these divaricatus or woodland sunflowers with Annuus pollen, pollinated thousands of heads, and the only seed we ended up getting out of that effort was selfed divaricatus sunflowers. Yaniv, do you want to say anything else about that?
Yeah, it's not that exciting, but yeah, we spent a lot of time. We thought we had some success. So it wasn't just the self divaricatus, it was also, there were some polyploid divaricatuses that's called Hirsutus, I think.
Yeah.
And those crosses worked as well. And then we found out they were polyploid. And so then we were back in the same, issues with fertility that Jill was talking about. So this was based on some preliminary work or some old published papers that people said this worked in, but presumably they had the wrong, they probably had the polyploid divaricatus when they did that analysis. So we couldn't get this cross to work. So that was
so you can make seeds from the 2, but those seeds are sterile, I guess, because they have triploid pollen. Okay, so this is just, so as far as the crosses in this, we're just doing, it's just a Novo Domestication, no hybridization. So we're just crossing the different accessions. So we've done good by good, we've done different by different, and I plan to plant a large cohort of these crosses out in the field this spring. And then I'm currently working on crossing plants for genetic diversity. So in the woodland sunflower, we're aiming for larger seeds and heads, but we'd also like to see fewer branches and a preponderance towards a large central head, rather than the wild type in which there are many heads that are all the same size. There is variation in this trait, and this is just a picture of one of the larger headed plants. And this graph here is showing the seed weights of the conspecific hybrid seed in blue. And then the yellow line through the center is actually composed of individual dots, each of which represents the midpoint weight for the parents of the hybrid in the same vertical slice. So any blue dot that's above that yellow line represents a hybrid seed weight that's higher than the average of its parents. And this dashed line that I just put up is, it just shows the heaviest seed weight for any individual accession. So, so you can see that many of the hybrid seeds are heavier than that. So that may be an early sign of hybrid vigor. And I did plant a few of those seeds out this summer, a little later than would be ideal, but hopefully I can measure some of them next year. So this summer, I've been collecting a lot of phenotype data on these accessions, including phenology, which is shown here. This is just a box plot, so it's showing the spread of the data for each accession in quartiles. And you can see that there's quite a bit of variability between the accessions. Y on the far right, that is composed of the entire population of mostly open pollinated conspecific hybrids between the accessions. So it makes sense that the flowering dates for that group span the entire range of flowering groups, but there are large ranges also within a couple of the individual accessions. So, so yeah, high flowering time variations within and between accessions in this species. Here are some floral and morphological measurements that I've been collecting, again showing the spread of the data for each accession in quartiles. And the main thing I take away from these figures is that, as with the previous slide, there's plenty of variability in these accessions. I am particularly interested in this accession 435675 for its higher potential for seeds per head. So moving forward, we intend to keep phenotyping any new material. In particular, we'll be looking for crosses that produce those agronomic features I talked about. And on that note, I'll mute myself so Kevin can tell you what he's been up to with our hybrid population. Is Kevin there? I see Kevin. Kevin, you're muted.
Okay, how's that?
That's better.
All right. Okay, I'd like to update you on some preliminary stuff that I presented earlier this year. I have some data to fill in some of the blanks that for the questions that I was asking earlier.
Let me introduce Kevin. This is Kevin Betts. Kevin Betts has worked on the Forever Green project from the very, very beginning. And this is the individual that is basically has been responsible for the early stage development of a large number of the Forever Green Forever Green crop. So he has maintained a focus on the perennial sunflower because he thinks he can make it work someday. So he's been very, very committed and has carried a very large portion of the role in the development of the perennial flower sunflower program in partnership with the rest of the team. So I just wanted to say that before Kevin started.
Well, thank you, Don. Yeah, I would like to have success in this before I retire, and I'm kind of running out of time. So hopefully we make some progress really quickly here. But I'd like to just show you where we are now. So if Jill, if you could flip the slide, please. Little background, the original cross, as Jill mentioned earlier, was between Jerusalem artichoke and domestic annual sunflower. And that resulted in interspecific progeny that were tetraploid, but varied in overwintering ability, flower number, flower size, seed size, and pollen fertility. Next, please. Since the original hybridization, selections in each generation have been made toward the ideotype of a large single central flower per shoot. The bigger the size, the better. 10 centimeters was a goal that we set. And we'd like the plant to be a tuberizing plant that would reliably survive our winters. And we need, of course, vigorous production of viable pollen and hopefully stable pollen, and not like some of the slides that Jill had presented earlier. Like to have a large seed producing approaching 5 or 6 grams per hundredweight, and which would be comparable to an oil seed, crop oil seed on the smaller end. And of course, high percentage seed set. Ideally, I'd like to have self-fertility. That allele was discovered quite early in sunflower development, and it actually made possible the use of sunflower as a crop plant, where you perhaps have one really ideal line planted in the field, and it wouldn't need to have the ability to outcross. And in the wild state, of course, sunflowers are self-incompatible. So if you had a large field of genetically similar plants, you might not get good seed yield just due to the fact that they'd be incompatible with like individuals. Next slide. These are the lines that I consider to be the elite lines that we have right now. I have for convenience, I've labeled them A through J, but the flower size is in all of these is between 40 and 65 millimeters. The ploidy estimate indicates that they're pretty close to tetraploid. The pollen fertility, where we have some data, appears to be quite good. As far as the perenniality goes, all of these were selected because they have exhibited overwintering, at least at one point or another. The fourth one on the list, D, is actually the one that I am the most excited about. This one is strongly perennial, but it still does have large flowers. Then we get to the seed production part, which has been kind of the bugaboo throughout all of this. They are all pretty self-incompatible. When they are allowed to outcross, you do get some seed set. But for flowers of this size, I did a calculation. For flowers of this size, I would expect 400 to 500 potential seeds off of each of these flowers. So you can see we're well below that. And that's what we'd like to attack with this selfing idea. Next slide. This is to show you what these ideotypes look like in real life. This is Now, we can go through these fairly quickly. Next slide. And the perenniality is decent in A, and these are the rhizomes that it produces. Next slide. B, which B actually has the best flower characteristics of any of these, so that it's good in that respect. Next slide. Here's the overwintering structures here. The rhizomes are a lot less developed than some of the others, and as a consequence of this doesn't overwinter very well either, but it will overwinter in a mild winter. Next slide. D is the one that I'm the most excited about. The flower size is a bit smaller, but it seems to produce excellent pollen. And next slide. The rhizomes are very strong. It produces several new shoots every spring. So in terms of overwintering in Minnesota, this is probably the most exciting candidate. Next slide. So after several generations of selection and crossing of these desirable phenotypes, we now have some plants that have good overwintering ability, and the single central flower per stem is present in all of these. The flower size is improving, and the pollen production of all of these, at least outwardly, appears to be quite good. The seed set of these lines remains very low though. So in order to overcome that seed set issue, I wanted to try a recurrent self-pollination strategy. And hopefully this is something that we really haven't investigated super intensively over the past years. And it may turn out that the meiotic difficulties that we see in the pollen slides that Jill had may be the largest factor in the low seed yield. But one thing I did want to rule out was the fact that, the wild type allele for seed set is toward the self-incompatibility end. And if it were possible to introduce and amplify, a self-fertility allele in these lines, maybe that's a significant issue to our seed set, but that remains to be seen, I guess. Next slide. And my rationale for why this may work is that the initial hybridization with annual sunflowers should have introduced the self-fertility allele into this population. So it should be there. And, wild sunflowers, including artichoke, the wild type, of course, is self-incompatibility. And that's going to be the most commonly occurring form, even in this population. The current lines likely carry the fertility form of the allele. And by using recurrent selfing and selection, I'm hoping to amplify that allele within the entire genome and maybe improve the seed yield. I did in very simple terms, I did a gene frequency calculation, and if everything was all the appropriate chromosomes were homologous and everything like that, at the end of the second cycle of selfing, theoretically there would be one plant out of every 36 that should show a massive increase in seed set. Next slide. Of course, things are not simple like I would like. The original perennial parent, the artichoke, is probably an allotetraploid, which means it has, two genome sets. The A set, which is probably from the perennial ancestor. The B set was possibly from an annual ancestor. So it may be that the chromosomes are not homologous and won't cross in a very predictable way. And if that's the case, then it may not be possible to amplify the fertility gene as much as is needed to get the sort of boost that I would like. You go to the next slide. These are the rows of my ideotypes, and you can see they're pretty uniform in size, and you know, if these plants actually produce seed like I would like, they would be at sort of the threshold of being able to release as a crop. But of course, they, at best, when they're outcrossing, they maybe produce, you know, about 8% of the potential seed yield that they should. Next slide. So anyway, the idea was to take some of these ideotype plants and self them recurrently. So for the most part, I have self ones growing in the field right now. I have a very few self twos that for whatever reason I had self the season before, so I had a little bit of data on those too. But what I would like to see, of course, is an increase in the self ability of these plants over generations. Now, if this were what I would, you know, if this were an ideal world and it were working exactly the way I wanted, I would see some increase in the self ones in terms of seed set. By the self two, I would like to see a massive increase. Yeah. So looking at D, for instance, which happens to be my favorite plant, at the start here, we have 7 self seeds per flower, and that represents the best plant I had. You know, I had plants with 7, 6, a lot of zeros, ones, like that. So in the self one, after one self, the best flower I had 16 seeds per head. Then, and without crossing, you get a little bit higher than that. So, you know, ideally at the end of the self 2 generation, I'd like to see a real big leap, and it incrementally increased. It's up to 38, but that's not really the huge jump I had hoped for. But as I said, the set of seeds that have been through 2 selves is not very large right now, and if my calculation of 1 out of 36 being sort of the frequency of something that really leaped forward, maybe I didn't have enough plants there. But that's something I'll discover in the greenhouse this fall or this winter. I have a lot of S1s now that I will self again in the greenhouse, and hopefully out of that whole batch of stuff, there will be something that really takes a big jump. But at this point, I'm not seeing it. But I would really like to rule this out. It would be really nice if this was the major issue with seed set rather than uneven gametes and stuff like that, because this is something we could deal with. Next slide. And one of the other things that I was hoping wouldn't pop up quite this strong, when I started selfing them, I got a lot of variability of the progeny, and that was not necessarily to the good end. A lot of them were less vigorous. There were still some that were quite desirable, but a lot of them turned into garbage. So that's something that is not necessarily a problem as long as, you know, a few good ones remain. But with each generation selfing, a lot of stuff seems to fall apart. So that's another thing I'll have to wade through, I guess. Next slide. This slide is sort of illustrates the variability that comes from a single self flower. Now these were all from the same flower, and yet the plants that were derived from these seeds were quite different in almost every respect. And the encouraging part, I guess, is that along with the bad, there were some that were actually really good. So it's almost like starting over in a sense that each flower produces a whole range of phenotypes, some good, some bad, and you know, hopefully that would stabilize as I continue selfing, you know, good plants. So that's my hope anyway. Next slide. And these are some of the better plants that I saw in the self one generation. So next slide. This is a self from one of the ideotype Ds, which is the one that I really like. And this one maintained a good head size, and I'll have to, you know, data is still being collected as to the perenniality of it and that sort of thing. I'm gonna have to go out one more time this year and dig rhizomes that I can bring into the greenhouse. But this one looks a little bit promising. Next one. This is a self from ideotype B, and this looks similar to B in a lot of respects. The seed set appeared to take a little bit of a bump upward, and that's certainly what we want. Next. And this is a self from ideotype C, and again the seed set again appeared to improve slightly. Next. So going into this winter, I'll be harvesting the self seed and threshing, which I'm doing right now. And also I'd like to bring rhizomes in from the fall of selected plants, and I'll grow those in the greenhouse, and we'll do another selfing generation in the greenhouse, hopefully, and have some seed ready to plant into larger plots next year to screen and look for improvements and throw out bad stuff. And hopefully, you know, we'll see some real positive stuff coming out of this. And but I probably won't know that until I see what the greenhouse stuff looks like this year. So so that's good and bad. I think, you know, after the second selfing generation, I think I'll have a pretty good idea if we're on the right track or not. So if there's nothing in there that looks like it's really substantially better by the end of the second self, I think we'll know that this is maybe a dead end, but hopefully it won't be. I'd really like to succeed in this before I retire. So that's all I have for today. So any questions you might have for either of us?
Well, let's open it up for comments and questions for the team.
I have a question.
Go ahead.
First of all, nice presentation, great progress. And Don, like I talked to you, I had a dream for perennial crops. So it looks like my dream is coming closer, closer into true. So
but it's not
probably,
easy.
yeah, it's not easy. And my name is Zhang Yuxu. I'm an alfalfa breeder. The background of my question is alfalfa is a perennial. It can be used about 4 years or maybe longer, but first couple years with high yield and then the decrease. And relate to
You're talking
that
about
is,
forage yield, right?
yeah, the alfalfa. So can you hear me, Dan?
Yeah.
Okay, so I had this idea is if we can double the alfalfa usage of year from 4 to 6 or to 7, then that would be great. So relate to the background of my question is when you make a selection for the perennial sunflower, what is the range of the year variation from the population? How will you select longer year than the parental line? What is the target year and the practical number of years we use? I know this is a later stage question, probably since we have here, you have made huge progress on the morphological trees like seed size, fertility, etc. But in long run, if we think 10 years later for the perennial sunflower in the field, then what kind of trait we need to improve? If we can think about the question at the beginning, probably we can benefit from that. So that is my question. I'm not sure I explained myself.
Well, but what's your question? How long will these survive? How many seasons?
Well, like the alfalfa we can use 4 years, and then
Yes.
we need to replace a new one. And
Yeah.
for perennial sunflower, and what is the variation of number of years from the segregation population or from the breeding population? And
Well,
from those,
yeah, they're not as long-lived as alfalfa for sure. The best good plant I have has been in for 3 years. A lot of times they will survive 1 year and overwinter one time, and then they maybe don't make it the second time. So I don't have a lot of good plants that survive many years. The ideotype D has been in the ground for 3 years and survived. So that approaches the, I mean, I think by 4 years, I would agree with you. In my experience with perennials, they do lose productivity after 3 or 4 years. And, you know, if this were a crop, I'm sure we would just rotate it to something else probably.
yeah. So what is, have you noticed any progenies that can live even longer or more years than the parental line?
Well, yes, there are some that have been in the ground for probably 8 years, but they're not desirable types in terms of seed production. The true perennial ones have small flowers, and they stay alive almost indefinitely.
I see. Thank you.
Yeah.
The ones that I work on, the woodland sunflower, they can stick around 4 years or, you know, or longer than that, but they again are, yeah, they're a wild sunflower and with much smaller seeds than our other project too. Yeah.
Well, this is great work. I can see the potential to change the world. So I stay in Inner Mongolia about 7 years when I study over there. And the grass land is becoming desert because they because probably they harvest the grass too frequently. People right now to plant the sunflower. If we can have perennial sunflower, the root will benefit the soil dramatically. So probably I'm not a sunflower breeder, but just think about that relate to the grassland and the farmland over there too.
Kevin and Jill, this is Carmen. Have you done work on the impact of climate? You know, we do have the much colder climates here. Does climate seem to be a significant impact in, you know, keeping this thing from really taking off?
Climate in what sense? Our cold climate? Yeah.
Yeah.
Yes, it does. So, a lot of the plants that I have that actually produce, you know, have decent sized flowers and everything are right on the margin of surviving, because some winters they make it and some they don't. And that's why D was so exciting to me, because it seems to make it every year and it seems to be stronger every year, no matter what the winter is like. But the other ones are kind of more marginal in that way.
Thanks.
Yeah, I would say that we had a pretty rough winter. The winter before last winter, where Kevin and I both saw higher mortality in our fields. So yeah, I mean, a rough winter is going to be harder on perennial crops too.
But the point in this program is that you have identified an ideotype that does have a level of winter hardiness that can be as the base to continue the program off of, right? You know, you've shown proof of concept.
Yes, in fact, I would. Yeah, I would call that one very strong in overwintering, which is good. And the other ones, I really can't say that they overwinter okay, but you can't really rely on them every year.
Just a crazy thought or a crazy idea. Have you thought about planting a cover crop in the late fall to provide some cover protection, snow catching capabilities, and if that would impact?
Well, I haven't thought about it while they're in the nursery stage now, because of course, I don't want to make life easy for them. When you're doing the selection, of course, you don't want to make, grease the skids for them. Really,
I hear you.
You want to make,
okay.
Yeah.
Mikey, did you have something to say?
Oh no, I was just going to say that there was the question in the chat that that was asking about bio or phytoremediation. And that we I just wanted to remind people that we haven't tested it, but it probably would be very fun too.
But just to remind everybody, there's all kinds of questions that can be asked, but we need the real plant material first, right? to go through those issues that these guys are raising in terms of fertility and winter hardiness. Has been and will continue to be, obviously the focus of your guys' program, right?
Don, this is Rod Larkins. I think I know the answer, but funding is always nice to drive these programs, and we're currently scouting for some new LCCMR partnership opportunities. This strikes me as being maybe a little premature to start thinking about an LCCMR, but I thought I'd at least ask the question.
Well, it'd be up to this team, but as you well know, that's the reason we have these lab meetings once a week for 2 semesters, is to bring everybody up to date as to where everything is at, and and kind of have a group conversation about how and where we should, search for the funding. But we want everybody to know the exact details of where everything is at, right? So you can see today where this program is at. Those two issues have to be dealt with before, this was going to be getting close to going out into the landscape. And Kevin showed you some nice uniform lines, right, that have the potential, but he probably won't know, the team really won't know where they're at for probably another 2 years, I would guess, right, in terms of whether something would be ready to go out for some type of field testing in one of our cluster regions in the state where we're advancing with you guys at ARI, the development of the supply chain and production and supply chain work. So I would say my take would be it's a little bit early for that.
Yeah, that was kind of my thought, Don.
Yeah.
Well, relate to that, Kevin, you talk about the retirement. What is your guess, the number of years, optical number of years, optimistic number of years needed in order to put into the yield testing, not in the farmer's field, but in like experiment to test the real yield, whether they're comparable to the annual sunflower. How many years do you think we need
How many?
to do that?
That would just be a guess. If we're able to improve the seed set, then it wouldn't be very long at all. But until we improve the seed set, what we have now is not going to compare to annual at all. It just doesn't produce enough seed. But I mean, theoretically, if each flower produced all the seed that it should, then it would be, it'd be appropriate to do sort of a yield trial. And I don't think it would yield as much as an annual sunflower because the flowers are smaller, but each plant would have one flower per stem. So overall, when you add all the flowers together, I mean, theoretically, it could come close to annual sunflower. But until we have something that produces seed reliably, it's going to be premature. If we found even one plant that was self-fertile and it had good seed set, we could start yield trials, probably 2 years after that plant was discovered, I would think.
Really good. Thank you.
Yes.
John
Sorry.
Hill.
I had a quick suggestion, but I didn't want to step on anyone else's question. So Kevin, you spoke briefly, I want to go back to something that was brought up earlier about the idea of planting cover crops to help with winter survival. And Kevin, you said you didn't really want to make it too easy for them in the breeding nursery, but I know at least in silphium and other crops, we're thinking about this idea of breeding for systems. I wonder if
right.
Planting a cover crop is what it takes to get a high yielding perennial sunflower in Minnesota, that maybe targeting that system and presenting that as a kind of a holistic thing isn't a bad idea.
Well, I can see that, but I mean, it's sort of like, my major focus is just getting seed set right now. And if I had a plant with seed set, then we would still be able to do some selection in sort of a more complex environment like that. But until I get seed set, we have nothing. So I guess that's why I focus entirely on that. And I do. I do have to make sure that I'm screening as severely as I can for winter hardiness. And, cover would probably, help them out a little more than I want to. So at this point, I see your point, and it's a good point, but I would like to have a plant that's productive before I start doing that kind of selection, I think.
That's why Kevin's nickname is Killer.
Then, nice job, team. I'm sorry I have to go to the next meeting, but we're done.
One question I'd like to ask you guys, you guys have, and as I've watched this work progress, we made the decision years ago that it was going to be a one flower plant. What if you had a plant that had two flowers that matured at the same time? What impact would that have on yield? I guess what I'm asking, should we continue to just look at that single plant, a single flowered plant as an ideotype, or are there some other possibilities?
Well, when I say single flower, really they have one flower per stem. So, the plants that I'm showing you have, they probably have 10 or 20 shoots, and each one of those has a flower on it.
Yeah.
They don't branch on each stem is all, but they kind of
are
are
they-
what describing though anyway. So
What's the uniformity in flower maturity when you have multiple stems?
they don't vary by a great deal. It's probably 2 weeks.
Yeah.
But I mean, if you're letting them go to maturity, there's a time frame where they're all pretty much mature and you could combine them. Yeah.
Yeah.
Well, and Kevin, if you have that 2-week variability in maturity, is there any concern about crop loss? I mean, like in currants, we're talking about shattering. How about in sunflowers, your experience?
Well, I mean, yeah, sure. There's always bird feeding going on, so that would be an issue for sure. And some of these have they select the types that face these don't. They're more of the easy for birds to spawn orientation, and that's probably not so good. But so there's issues like that for sure.
So, are there any other comments or questions?
Yeah, Don, this is Rod again. I was just wondering what the relative size and weight of the seeds are of the perennial versus the annual black oil seed that you see.
You guys showed some of that data, right, Jill? What is it? What is the difference in seed size? You showed the picture of it, but what is the?
Well, I don't have the weights. I have Kevin's been taking data on seed measurements. So if I go back to that.
I think it was, yeah, I think, crop sunflower is about 6 grams per 100 weight, and this is probably, a third to a half that. So they're lighter seeds for sure.
But if I remember the conversations earlier, that some of them were about half, right?
Well, yeah, in gross size they were about half. By weight they're probably about a third as much.
Okay.
As an annual.
Yeah. But that's a trait. Tell me again. That's a trait that you can breed for. You can advance or not.
I think so. Yeah.
Yeah.
No, I see there's a lot of range in all of this stuff, and some of them actually are indistinguishable from a small oil seed, but
yeah,
again, if they don't produce seed, that doesn't count. So
yeah, right. Well, Kevin, it appears that we're going to retire together when we're 100 years old.
Well, I didn't say I was going to wait till I retire. Wait to finish before I retire. That will probably come first. I don't know. But I'm motivated. I'm motivated to get this done in 2 years, though, for that reason, because otherwise.
Well, again, thanks to the team, and then I was speaking on behalf of the team, all the work that Kevin has done to keep this program running as the students come and go, and putting this team together now. Just want to again thank Kevin for all of the field work that he's done in support of the program. So,
all right. Thank you,
any
John.
other comments, questions? If not, thank you for the great update today, team, and for everyone that's still in line, we welcome you back every week at noon for a continued update of the Forever Green Research Project. So have a great weekend. Stay safe, stay healthy.
Thanks, Don. Thanks, everyone.
Thanks, Don. Bye.
Thank you, Jill.
Yeah, thanks, everybody.
Bye-bye.
Bye.